Most of us will need or use a drug at some time in our lives. Drugs come in many forms, from disease-preventing vaccines and insulin injections for diabetes to inhalers that prevent or relieve asthma. Shops are full of remedies for coughs and colds, as well as aspirin, paracetamol and ibuprofen for the relief of mild pain and fever. You're likely to have heard of opiates like morphine from poppies being used to treat severe pain. And there are now many types of chemotherapy for cancer and microbial infections. Specific milestones are reached along the pathway to drug discovery. The process begins with deciding on which disease to treat and identifying a molecular target which, when modified in an appropriate way, will have beneficial effects on the disease. That involves extensive studies to identify any genes that might be involved, to define the pathology of the disease, and determine how physiological and biochemical processes are altered in the disease. Once a target has been identified, it is validated before progressing further. That involves experiments to confirm that modifying the target does indeed produce the effects that you predict from what you have learned about it. Once a molecular target has been identified and verified, the search for a new drug begins by looking for molecules that can bind to the target. Many molecules will bind but have no effect, so are not any use as drugs. So, any compounds that bind are tested further to determine if binding to the target results in an effect. Compounds that bind to the target and produce the desired effect by the intended mechanism are called hit compounds. These molecules move on to the next step in the drug discovery pathway. Some of the molecules identified as hits might have properties suitable for use as a medicinal drug, but most will not. Further tests are therefore done on the hit compounds to assess their biophysical properties and their activity at and selectivity for the target. They are also investigated for potential pharmacokinetic problems, including how well they would be absorbed by the body and how they might be metabolized. A major aim in drug discovery is to identify compounds that are safe, so the compounds are also assessed for their potential to cause toxic effects. Compounds don't need to be perfect at this point, but they must have structures that are amenable to being tweaked by medicinal chemists to make them more drug-like. Molecules that fulfill certain criteria are considered potential drug candidates and can be taken further along the drug discovery pathway. These compounds are known as leads. At the next step, lead molecules direct the search for a drug candidate. Several series of molecules with structures based on lead compounds are synthesized. They are then tested in a series of assays to assess a range of properties. Depending on the outcome, the structure may be repeatedly modified and the compound retested each time in an optimization cycle that hopefully leads to a new drug. The aim is to make a molecule that has the active moiety of the lead, but is better absorbed by the body, is more potent and more selective for the target, and lacks any toxic groups. This process is called lead optimization. Once you have identified a molecule with the desired pharmacological activity and chemical properties, it becomes a candidate drug. And the next step is to evaluate its effectiveness and toxicity in a wider range of assays. Drug candidates now reach the stage of preclinical testing. Up to this point, the testing of compounds relied on chemical and biochemical assays and in vitro cell-based assays. To truly understand how a drug will work when given to a patient, it is essential to study it in systems that replicate the environment it will be used in. This involves in vitro testing on human and animal cells and tissues, as well as in vivo studies on animals. This part of the process is highly regulated by national and international agencies that set standards for ethics, study design, animal care, and reporting of results. The aim is to ensure the safety of a new drug before it can be tested on humans. It's only after detailed information has been gathered to ensure that the new drug is likely to be safe that it can be administered to humans. The first point at which a drug can be given to humans is in a clinical trial. These trials are highly regulated by international authorities, including the Food and Drug Administration in the USA, the Medicines and Healthcare Products Regulatory Agency in the UK, and the European Medicines Agency. The International Council for Harmonisation of Technical Requirements for Pharmaceuticals for Human Use, or ICH, develops guidelines for clinical trials, and the World Health Organization provides guidance on ethical and scientific standards. These regulations are in place to maximise the safety of the first people to take the drug. As a result, clinical trials are generally very safe. They take place all the time, and it is only on the very rare occasions when something unexpected happens and someone gets hurt that you hear about them. There are 3 main phases of clinical trials that must be completed before a drug can be approved for use in humans. Each phase must be passed successfully before progression to the next phase is allowed. Drugs are usually tested first in a small group of healthy subjects in phase 1. Phase 2 is the first time a new drug would be given to the intended patient group, and it is only given to a small number. This is when you find out if the new drug actually works. If a beneficial effect is found and it outweighs the risks, the drug may then be able to progress to phase 3, when it is tested in a large cohort of patients. Once a new drug has successfully passed all the required stages of clinical trials, a new drug application, or NDA, can be submitted to the regulating body. The NDA includes data from the preclinical and clinical studies, information on the manufacturing process, and proposed labelling for the drug. The agency reviews the data submitted and considers the safety, efficacy, and quality of the drug, especially the balance between benefit and risk. If approved, the drug is authorised for marketing. After approval, the drug's safety and effectiveness continue to be monitored through post-marketing surveillance programs in what is known as pharmacovigilance. Drug discovery is a complex and slow process, taking on average 10 to 15 years from the initial ideas about a molecular target to getting a drug approved for sale. The initial steps are the fastest, but once a drug has been proven to work safely in preclinical trials, it can take many more years to prove its safety and effectiveness in human patients. Of the drugs making it to clinical trials, more than 80% fail the testing and never become a therapeutic drug. In phase 1, where the main focus is on safety, about 10 to 20% fail on average. In phase 2 trials, 30 to 50% fail, mainly because they don't have the intended effect when administered to patients. About half of the new drugs getting as far as phase 3 trials fail, mainly because they are no better than older drugs, or there is an insufficient overall profile of benefits versus risks. Even after a drug has been approved for marketing, it can be withdrawn if concerning side effects are encountered. But this is rare. The long and rigorous processes followed during drug discovery and development mean that drugs making it to market are safe and effective. But rare side effects can occur, but may only be seen when the drug is used by very large numbers of patients.